Hello everyone, myself Sakshi Dinesh Patel. Today I am going to present on the topic mineral oil overlay method for microbial preservation. In this presentation, I am going to explain what is preservation, what is the method, procedure, requirement and application, advantages and disadvantages of microbial preservation. Introduction to microbial preservation. Microbial preservation means maintaining microorganisms in living and stable condition for their future use. It is important because microorganisms can lose their characteristics and viability if they are not stored properly. Preservation method helps in preventing contamination, dehydration and loss of important properties. There are different types of preservation methods such as refrigeration, freezing, lyophilization and mineral oil overlay. Now we will move to next slide, which is mineral oil overlay method. Mineral oil overlay method is a simple and economical technique used to preserve microbial cultures. First, the microorganisms are grown on a suitable agar slant. After sufficient growth, the cultures are covered with sterile mineral oil. This oil forms a protective layer over the culture. It helps reduce moisture loss and limits exposure to oxygen, which slow down the microbial activity and helps in maintaining the culture for longer period. Next slide is of principle. The principle of this method is based on creating a protective environment around the microbial culture. Firstly, the microorganisms are grown on agar slant. Then the sterile mineral oil is added over it. Then the oil layer reduces the moisture loss and oxygen exposure. As a result, the metabolic activity of the microorganism is reduced. This helps the microorganism remain viable for the extended period. The simple example we can see that is the mineral oil is a protective covering that helps prevent the culture from drying out. We will move to next slide, which is material required for this process. The main material required for this method include a pure microbial culture, suitable agar medium, culture tubes, sterile mineral oil, an inoculating loop, sterile pipette or syringe, incubator and autoclave. It is also important to maintain an aseptic environment throughout the process to prevent the contaminations. Now I will explain the procedure of this method. Firstly, a suitable agar medium is prepared and sterilized. The medium is then placed in culture tube to form an agar slant. A pure microbial culture is inoculated onto the agar slant using aseptic techniques. The tube is incubated until sufficient growth is obtained. After growth appears, sterile mineral oil is added over the culture surface. The tube is then closed properly and stored under the suitable condition. When the culture is needed, it can be recovered and transferred to fresh medium for future growth. The next slide is of advantages and limitation. In this method, there are several advantages. It is a simple, economical and require minimal laboratory equipments process. In this process also helps in reducing dehydration and slow microbial metabolic activity. However, it also has some limitations. It is not suitable for every microorganisms. Some cultures may also lose viability during prolonged storage, and genetic changes may occur over the time. Proper aseptic handling is also necessary to prevent the contaminations. Applications and precautions. The mineral oil overlay method can be used in research laboratories, teaching laboratories, industrial microbiology, biotechnology laboratories, and for maintaining microbial strains. While using this method, we should take certain precautions. We should use a pure culture and maintaining aseptic conditions. The mineral oil should be properly sterilized and the culture should be completely covered with oil. Proper storage conditions should be also maintained. The next slide is of references. The information present in the presentation was collected from the reliable scientific and microbiological sources. The main references include a review article on microbial preservation method, the American Type Culture Collection Microbiology Guide, and a research article on long-term storage of microbial cultures. At last, I like to conclude that mineral oil overlay method is a simple and economical technique used for preserving microbial cultures. The mineral oil acts as a protective layer, reducing moisture loss and oxygen exposure, which helps maintaining the viability of microorganisms. Thank you for listening my presentation.